Okay. Let's close this. Okay, good afternoon, everyone. Welcome to BMS 300, Principles of Human Physiology. If you're not here for Principles of Human Physiology, now is a good time to discreetly leave the class. I'm Dr. Michael. I'm the course director, and I'd like to begin each lecture with an overview of what we'll be discussing. Right. So the first thing I want to do was just make sure you are sitting with your TAs, and that's why I had this slide up there. But for now, just find your places, and if you can always find your TAs later as well. So here's an overview of what we'll be discussing and doing. I hope you've already found your TAs. If you haven't met them as yet, just head on down. We have a number of open seats down here as well. And then I'd like to introduce you to the teaching team. So this class has two sections, one at 11 a.m. and then the 12 p.m. section. And in total, we have over 560 students, right? So since we have such a large class, we need a large teaching team as well. And so I'll introduce you to the main instructors, and also the grad TAs and the undergrad TAs as well. And you would have also noticed that there were these scantrons which were handed out at the start of class. I just want to reassure you that you're not having an exam today. What we're going to do is a pre-course survey where we want to see how well the prerequisite courses have prepared you to be in BMS 300. All right, so this is more a test of the prerequisite courses and how much you remember from that, and not really a test of you guys. All right, so please go ahead, write your names on the Scantron and also the student ID. If you haven't done that already, please do so, because without knowing your name and ID, the data we're collecting will be pretty useless. All right, and TAs, could you just hand Scantrons to a couple of people at the back? If you don't have Scantrons, please raise your hand and you should be getting a Scantron at the earliest. And then what I'd like to do is give you a content overview where we'll have a bird's eye view of what will be discussed in this course this entire semester. And then we look at the mode of assessment and how the delivery will be as well. Right, and I'll just briefly walk you through some of the key features in the course website. So I'd like to start by telling you about the teaching team. So we have two grad TAs. We have John and Addy at the back. There's Addy waving and John talking to Gabby. Right? Yes, they'll be there. Yeah, they love to meet students, and they are all very excited to work with you. So the reason we have TAs in this class is because when you have over 560 students, it's easy to get lost in the crowd. And I was telling the earlier class that I don't claim to be as engaging as a movie where they're all just fixated on me. So we like to have breakout sessions in between where the TAs will be facilitating a group discussion as well. And the thing is, the reason we decided to have these TAs is for a number of reasons, and we want to improve the overall class environment. So historically, when I was sitting at the back and watching my co-instructor teach, I noticed a number of things going on in classes, like people taking selfies, or online shopping. That's quite common in big classrooms, watching videos. And I'm not against any of this, right? As long as it doesn't happen in this room here, I'm totally okay with it, right? So we want to ensure that the environment is optimal for learning, right? Sometimes someone may be very capable of listening in one ear and online shopping, but the person sitting next to them may not be able to focus, right? And so I have a little, actually a little bit about these TAs have been updated on Canvas. So if you go click on resources, the very first thing which pops up is meet the team, and so you can know a little more about your TAs and get to know them better. And another reason we have the TAs here is when it comes time to write letters of recommendation at the end of the semester, and I'm happy to do that. All I can say as the course director is your final grade, right? So and so got an A, so and so got a B, and that's all I can comment on. But I will be relying on the input from your TAs to tell me how you are in terms of participating in group activities? Are you a team player, right? How good are you in terms of communicating scientific information verbally? So those are nice things to have on your letters of recommendation, and so I would encourage you to get to know them and start planning ahead, right? So the undergrad TAs, they're also in charge of the recitation course. So BMS 200 is the supplemental course for those enrolled in BMS 300, and this is an optional 1-credit course which if you sign on, you will have a chance to get into a little more depth in certain topics, right? So they meet once a week for 50 minutes, and the various time slots are shown in this table here. And if you are interested in enrolling, please use this course registration number, the CRN listed next to the recitation number. So we have a total of 8 recitation sessions. So there will be 2 sessions happening simultaneously from 1 p.m. all the way to 5 p.m. So you'll have 8 recitation sessions. And please register for this through RamWeb. And we found that historically those who take part in these sessions seem to have a better overall performance, because just having more repetition is always beneficial. And this is graded separately. In order to get an A for this recitation course, all you do is show up on time, participate, and you'll get an A. But in order to stay enrolled in BMS 200, you need to be enrolled in BMS 300 as well. Okay, so here's a little bit about me. I'm Dr. Michael, and I came to the US over 15 years ago as an international student to Washington State University. Right, we're all in the same Pac-12 conference now. But when I came as an international student, do we have any international students in the classroom at all? Yes, hello. Okay, great. Yeah, well, do stop by and say hello. Others of you can as well, right? So when I came as an international student, one of the questions I was asked most often was when I introduced them by my first name. When I told them my first name is John, they said, "Is it a real Indian name, or did you pick up a fake American name to make our lives easy?" Right? And my response was telling them a little bit about my Indian heritage. So I would tell them that this was a name my parents did give me, because Christians in India would give names from the English Bible. So the name John, Jeshurn, and Michael all are from an English Bible. And so the reason it is English is because the British ruled us for close to 200 years, right? And so that's why English is actually an official language in India. And so I get to tell them about my Indian heritage, right? So at Washington State, I did research, and that's what grad students usually do. And my research work was in this condition called dilated cardiomyopathy, which is caused by mutations in cardiac proteins. And so here's a picture of me slaving away over this muscle rig. And after grad school, I worked at Cornell University for a few years, and there I did research based on behavioral approaches to combating obesity. Right, but at CSU, I don't do much research, and my main goal is teaching. So let me tell you how I got into teaching as a career. So for a number of semesters in grad school, I was a TA for biochemistry, and in the very final semester, I won this teaching award as a graduate teaching assistant, right? And so that made me consider maybe I should give teaching a shot, in terms of a career option. Little did I realize that I did have teaching running in my blood, because my grandparents on both maternal and paternal sides were school teachers, right? And then I went to Cornell, where I was a instructor for a couple of years. Then I got promoted to being a lecturer, and I was there for almost a total of 6 years. And during that time, I had an opportunity to teach a number of courses in nutrition, physiology, and also public health. And the reason I mention this is I want you to know how I approach human physiology, right? I think about human health not only in terms of the individual, but also from a population-wide perspective as well, and that's why I give you this introduction. So if you ever want to get in touch with me about anything at all, the email is the best way to stay in touch. And since we do have over 560 students, please CC the course email as well, so that you can get a quicker response. And while we're on the topic of sending emails, here's a reminder of how to write proper emails to professor. You don't want to be remembered for the wrong reasons, right? So please copy paste this and use this format while you send emails. And with that, I'd like to introduce my co-instructor, Michelle.
Good morning. It's not really morning, is it? I always say good morning, sorry. There we go. My name is Michelle Steros. I'm the co-instructor and course coordinator for this course. What that means is essentially I am the person you will be contacting most often when you have a problem or a question. Anytime you send an email that's not directly to Dr. Michael or to the TAs, will probably be answered by me. I'll also be sitting in the back all semester long, so please feel free to stop by and say hello. Happy to answer any questions that you have about the course. I've also had some research background, and I can talk to you about that. I could talk to you about some labs here on campus because my work was here at CSU. So I worked in a lab that did some neuroendocrine studying, so I looked at sex differences in development. And I've also done basic neurocircuitry research as well. So, stop by and talk about that if you'd like. So I ended up here as a transfer student in undergrad. I came here from Florida, and it was the best move I ever made, surprisingly. But I love it here, and so I've just stayed since. I did my bachelor's in biomedical sciences, and I took physiology, and I loved it, and I became a UTA for it. And then I came back and did my graduate degree here. I did a master's in biomedical sciences, and I was a GTA for physiology as well. And then once I finished my coursework, I just stuck around to keep TAing until they finally gave up and hired me, and now I'm an instructor for physiology. So I've been with this course for about 6 years now. I love this material. I love physiology. Please come talk to me about it. I'm very approachable, I promise. No one on our team will bite you. We all love working with students, so please come see us. Okay, so throughout the semester, we're going to have Poll Everywhere. That's what we're going to use. It's not for grades. It's just to give you some practice questions, and it's for us to kind of see as a class, are you on track with the material that we just taught, or do we need to go over it some more? So we're going to play a game today, just as a chance for you to get set up with Poll Everywhere. This is going to be 2 truths and a lie, and you can get to it from the QR code is probably the easiest, but you can also use the web address or you can text the number. So my 2 truths and a lie. My favorite park is Disney. I'll specify to Disney World since I'm from Florida. I paddleboard with my dog, or I have a library. Oh, we didn't clear it. I was like 41 already. While you're doing that, I'll just mention too, my office is in the physiology building. So if you guys were around last year or last semester, well, yeah, it was last year, and you know that the physiology building was closed. It has actually been reopened, so you can come see me in my office. That's also where the TA offices will be, in the basement, which is not as terrible as it sounds. We've actually got a nice little setup. All right, we'll go with that. So, most of you think my favorite theme park is not actually Disney, and you are correct. I tell everyone I have a theory on this, but first we'll just show you pictures. Okay, that's my baby girl, Molly. She is one of 2 of my cabbies. She is a 13-year-old rescue with no teeth who walks around looking adorable with her tongue hanging out, and she loves to paddleboard. That is Wizarding World at Islands of Adventure and Universal Studios. Of the 2, I actually prefer Islands of Adventure. It was the first major theme park I ever went to, so it's just my first love, I guess. So that's my theory, actually. If you went to Disney first as a child, that will always be your favorite. But if you didn't go like me until you were an adult, you're kind of like, okay, that was fun. I'm done. Back to Islands of Adventure. That is also my library. I have a lot of books, way too many books. My husband has put a cap on the amount of books I'm allowed. So any new books I'm supposed to like cull. So now I'm hiding them around the house. But I also have, I had to build a library for it. So I'm kind of proud of actually being able to do that. So yeah, that's me. Thank you.
Thank you, Michelle. I must add that being a course coordinator is a heck of a job because you have to coordinate with the course director, with students, with undergraduate TAs, with grad TAs, and all of that happens behind the scenes. So I'm very, very thankful for Michelle's role in the course. Okay, so my two truths and a lie. I went to my favorite restaurant over 100 times when I was a grad student at Washington State. Maybe wondering how did I afford that. Option B, I had no hobbies when I was in grad school. Option C, I painted a picture of a horse when I was only 3 years old. Okay, we have a lot of responses coming in pretty quickly. Okay, most of you think it's a lie that I had no hobbies, and you are correct, right? So I did go to my favorite restaurant over 100 times when I was in Washington State. This was a place called Gambino's. It's a deep dish pizza restaurant, and it was love at first bite. And here's a picture of me on my 100th visit with the restaurant staff. And I did have a couple of hobbies when I was in grad school, and so because I was in touch with a lot of international students, I had the opportunity to pick up Mandarin and Japanese, right? So here's me with a bunch of international students. And yeah, when I was 3, I did paint a picture of a horse, as shown here. So I wanted to go to art school, like my sister, and my parents said, nothing doing, you can become an engineer or a doctor. And so I do have an engineering degree, and my wife calls me a fake doctor because I have a PhD, right? So anyway, so now what we're going to do is we're going to do this pre-course survey, and I want you to be as honest as possible, all right? You getting a perfect score on this by looking at your neighbors is not going to help us. I want to know which topics I may need to spend more time focusing on, right? So the first question is, which of the following prerequisite courses did you complete before enrolling in BMS 300? Was it Life 102, Life 103, BZ 101, or BZ 110? So knowing this will really help us kind of score the courses, right? Not score you guys, but to see how students are doing if they took these courses. And maybe if you haven't taken any of these courses because you transferred from a different college, then please write down the name of that course on your Scantron. That will be very helpful as well. Okay, so the second question is also on the prerequisite courses. Maybe you took Chem 103, Chem 107, Chem 111, or BMS 260. Which did you take as a prerequisite course? And as I said, if you haven't taken any of these courses, just write down what courses you did take to be able to enroll in this. Okay, so with that, let's move on to the very first question. Which of the following bonds are responsible for the dipole moment of a water molecule? Which of the following bonds are responsible for the dipole moment of a water molecule? Is it hydrogen bonds, peptide bonds, nonpolar bonds, or covalent bonds? And if you don't know or don't remember, please select that option as well. Okay, moving on to the next question. The R groups that are in transmembrane proteins are most likely blank because of the hydrophobic nature of the phospholipid tails. So the R groups that are in transmembrane proteins are most likely blank because of the hydrophobic nature of the phospholipid tails. So do you think these R groups are polar? Are they positively charged, nonpolar, negatively charged? What do you think? Are they polar? Are they positively charged? Are they nonpolar, or are they negatively charged? The R groups of proteins, of transmembrane proteins. Can we move on? Yes? Yes, all right. The primary structure of proteins is not influenced by blank. So the primary structure of proteins is not influenced by peptide bonds, or is it not influenced by the order of the amino acids, by phosphodiester bonds, or it's not influenced by the number of amino acids. Which of the following does not influence the structure of the primary structure of proteins? Okay, moving on to the next question. Which of the following metabolic pathways indicate the use of anaerobic metabolism to make ATP? So which of the following metabolic pathways indicate the use of anaerobic metabolism to make ATP? Is it beta oxidation, electron transport chain, citric acid cycle, or glycolysis? Which of the following pathways is indicating anaerobic metabolism? Okay, moving on to the next question. A blank group linked to carbon 2 of the ribose sugar makes RNA less stable than DNA. So is it the hydrogen group, hydroxylic group, carboxylic group, or phosphate group? So which group linked to carbon 2 of the ribose sugar makes RNA less stable than DNA? Okay, moving on to the next question. According to the central dogma, blank is generated from translation. According to the central dogma, blank is generated from translation. Is it DNA, RNA, protein, or tRNA? Is it DNA, mRNA, protein, or tRNA? All right, moving on to the next question. Meiosis is a type of cell division that results in blank cells, each having half the number of chromosomes of the original cell. Meiosis is a type of cell division that results in how many cells? Each having half the number of chromosomes of the original cell. So do you get one cell, or do you get two cells, three cells, four cells, or four cells? Yeah. Okay, moving on to the next question. Enzymes that form phosphodiester bonds between nucleotides function in the blank. So enzymes that form phosphodiester bonds between nucleotides function in the blank. Is it in the lysozyme, cytosol, nucleus, or the endoplasmic reticulum? Where would you find these enzymes? Lysozyme, cytosol, nucleus, or the endoplasmic reticulum? Okay, moving on. In a blank mutation, all codons after the mutation are different. In a blank mutation, all codons after that mutation are different. Is it silent mutation, nonsense mutation, missense, or frameshift mutation? So after which of these following mutations will all the codons be different? Okay, moving on. Transcription always takes place in the blank. Does it always takes place in the endoplasmic reticulum, in the ribosome, in the cytoplasm, or in the nucleus? Transcription always takes place in the blank. Is it in the endoplasmic reticulum, in the ribosome, in the cytoplasm, or in the nucleus? Okay, moving on to the last question now. Multiple proteins can be made from a single gene due to blank. Is it due to 5' capping, due to alternative splicing, due to the 3' poly A tail, or due to ligation? So multiple proteins can be made from a single gene due to blank, due to 5' capping, due to alternative splicing, due to a 3' poly A tail, or due to ligation. So please make sure you've filled out your names and your student IDs and also the answers to question 1 and 2, because without that the data we collect from your scantrons won't be beneficial. So if you're finished answering your questions, you can just send the scantrons all the way to the end of your aisle, and then the TAs can come and grab it. Thank you all for your participation. Appreciate it. So what did you guys think? Was it hard? Too easy? In between? Okay. So I just want to say that regardless of how you did in this pre-course survey, I would really want you to focus on these questions for the upcoming exams, because some of these questions may show up on exam 1. And all of these topics on which these questions were based, they are from the prerequisite course material, and I will be covering it again in the first couple of weeks in class, right? So it's not things I'm going to hold you responsible for what I don't cover in class. I will cover it in class, and then you will be held responsible for it on the exam 1. And the reason I'm doing this is having a strong foundation is really crucial to build to build a further superstructure, right? If the foundation is shaky, the superstructure will fall as well. So now let's quickly go over some of the learning objectives in this course. There are 4 learning objectives, and the very first one is to demonstrate an understanding of cellular function. Repeat after me. Cellular function. Okay, that was pretty weak. Let's try that again. Cellular function. Okay, again, cellular function. Okay, all right. I have a 2-year-old and try to get her to repeat things. Anyway, the reason why I do this is a couple of reasons. Those key phrases I make you repeat after me, it might show up on the exam. It helps to reinforce these concepts. Secondly, it's very easy to fall asleep when just one person is talking the whole time, right? So I want to increase classroom engagement, and my inspirations are the mascots and football games, and also maybe volleyball games. They really get the crowd whipped up, so I'm trying to pull a page out of their book, right? Okay, so now the reason why we're talking about cellular function is that we need to understand what's happening in a single cell before we can apply to how multiple cells work together and eventually the human body functions. And the second objective is to appreciate that there are different organ systems which make up the human body, right? If you look at this picture here, you have around 11 different organ systems. And so among these various organ systems, we see that every system is made up of different organs. For example, let's take the urinary system, which functions to filter the blood and to produce urine. The organs there would be the kidneys for filtering the blood. You have ureters to move the urine into the bladder. It's stored in the bladder and then it is excreted out. So knowing the various components of these organ systems is very important. So I want you to know where these organs are located, what are their functions, and what are the anatomical terms as well. So this is the second learning outcome. The third learning outcome is to be able to explain how these different organ systems, they are going to be integrated and regulated. Right? Let's focus on the word integrated. Repeat after me. Integrated. Right? Again, integrated. And the reason we need to see how they are integrated is, let me give an example. Let's say, for example, there is a cut on your skin, right? And the skin is the largest organ in the body. It's part of the integumentary system. And so a cut on the skin, depending on the depth of the cut, there could be loss of blood. And if you have blood loss, that will decrease the functioning of the cardiovascular system. Less blood is pumped to the various parts of the body. The kidneys can undergo renal failure because the blood pressure is dropping very low, and depending on whether after the cut there is some infection, the immune system gets involved as well. So this is just an example of showing you how if you affect one organ system, you're going to affect the other organ systems as well. And we'll see how these organ systems, they will work together to maintain health and to prevent pathological conditions, right? And the reason we look at pathological conditions is that we never appreciate how well something works until something goes wrong, right? And an example from last year is that I never understood the importance of the springs in the garage doors until one Sunday morning we tried to leave the house and the door didn't work because the spring was broken. And because it was a Sunday, we ended up paying that guy $1,000 to replace it. We were super upset, but we had to pay so much. You could get it done for a much smaller amount, but it was an emergency, right? So anyway, the bottom line is you never appreciate how well things work until it falls apart, and that's why we look at a few pathological examples. And then the last learning outcome is to demonstrate how when there are problems happening at the cellular and molecular level, how it can have an impact on various organ systems. So again, let me give you an example of when something is going wrong at the cellular and molecular level, how it can affect multiple organ systems. So in the integumentary system, we'll see that there are a number of proteins, and there are these certain proteins which help hold your top layers of skin down, right? And if there is a mutation in those proteins, that means there's a problem at the cellular and molecular level, and as a result, the skin will start peeling, And if the skin peels, that means that it is now exposed to the external environment. You can have dehydration, loss of water. There could be exposure to pathogens, so there could be infections leading to sepsis. So again, this is an example of how a problem at the cellular and molecular level can affect multiple organ systems, right? So I won't give you specific examples right now because we are still at the bird's eye view level, but I just hope you can appreciate how the very first learning outcome of cellular function is now tying with the very last learning outcome, where the cellular function can have an impact on various organ systems. Is that clear, folks? Yes? Okay. So when you think about the organization of the human body, we find that our body is made up of several cells. These cells come together to form different types of tissues, and these tissues will come together to form various organs, right? So if you take the stomach, for example, you will find in it the 4 types of tissues which we will be discussing. And multiple organs like the stomach come together and form the digestive system, which is an example of an organ system, and multiple organ systems make up the organism. So shown here on the right hand side is the 11 organ systems we'll be considering, right? And we'll consider each of this in some amount of detail. We won't go into too much depth, but I want to show you how all of these systems are working together. And so this slide here is telling you how all of these organ systems are integrated and working together, and we will be coming back to this specific slide multiple times in the semester. So at the end of 15 weeks, we will be talking about the reproductive system, of how a sperm is going to fertilize the egg, and you get a zygote which can develop into a whole new organism, right? And so we end the semester by talking about what's happening in a cell, and we start the semester by looking at the structure of a single cell. And so in a single cell, the major component is water, and so tomorrow's lecture will be focusing on the properties of water and also how various substances can interact with water. So some are hydrophobic, meaning they are water hating. Other substances are hydrophilic, meaning water loving, and there are other molecules which are amphipathic. They have both water loving and water hating properties, and an example of that is seen in the phospholipid molecules which make up the cell membrane, right? So the cell membrane is made up of phospholipids. And so if you look at the cell membrane, you notice there are a number of proteins embedded in it. These are the transmembrane proteins. They're going across the membrane. They're transmembrane proteins, and your question will be, how in the world did they get here, right? So to answer how these transmembrane proteins get embedded there, we'll be talking about protein synthesis and how different organelles will play a role in protein synthesis. This also brings me to the concept of central dogma, where the DNA will give rise to RNA in the nucleus. The RNA is going to leave the nucleus in the cytoplasm, and there it is going to be translated to make proteins. So we will be talking about several types of proteins, but I want to focus on certain types of proteins which help form structures which hold cells together, right? So the structures which hold cells together are called cell junctions, and so we'll see how there are 3 types of cell junctions we'll be considering, right? Some will offer mechanical support, others are going to act as pathways for substances to move between the cells, and then other cell junctions, they just form a seal between the cells. Now when you talk about cells coming together, that will bring us to the concept of how similar cells coming together can form tissues, right? So as cells come together, they will form tissues, and some of them are formed by these cell junctions holding cells together. So among the types of tissues, we look at 4 of them. The first 2, even if you never took a science course, you would have heard about it: nervous tissue and muscular tissue. You will find that in the nervous system and in the muscles. But the other 2, connective tissue and epithelial tissue, is something which is maybe not as well known. We'll be talking about that as well. And so all of these tissues will come together to form various organs. So coming back to this topic of protein synthesis, we see that protein synthesis is expensive, and so it needs ATP to make ATP is required for making proteins. So where does this ATP come from? It comes from the macronutrients like carbohydrates, proteins, and fats. Where does that come from? It comes from the meals we eat. So the digestive system is going to provide the substrate required for making ATP. So when you think about the digestive system, it's controlled by the nervous system, and we can divide the nervous system functions into two parts. What is the voluntary action, like say picking up a bottle, right, and drinking the water. Involuntary actions like the production of saliva or maybe the gastric juices, all that is involuntary, right? So among that involuntary actions you have the fight or flight and the rest and digest response, and we will be seeing why eating is going to be optimal during the rest and digest response. The endocrine system also plays a role in the digestive function. How many of you have heard of the pancreas? Right? Yes, very good. Most of you heard of pancreas. So pancreas is an organ which will produce insulin, and that helps to maintain the blood glucose levels, right? Other endocrine organs include the adrenal glands, the thyroid glands, the neck region, and also at the base of the brain you have the hypothalamus and the pituitary gland. So these glands are very important even for reproductive function, because they will produce hormones which will act on the gonads like the testes and ovaries, and it can lead to the reproductive function. So let me tell you something really fascinating now. We're going to see how the reproductive function is linked to skeletal health, right? So how is the skeletal health influenced by the reproductive system? And the answer is, in women after menopause, the estrogen level starts dropping, and as a result the bone density decreases as well. And so you must have heard how after menopause females have an increased risk of bone fractures. And so we'll talk about that connection. Are there any things you can do to prevent this loss in bone density? Yes, you could engage in a proper diet and also these weight bearing exercises. And in order to do exercise, your brain will be activating the fight or flight response. Whether you're running away from something which is chasing you, or maybe you're just lifting weights, you need the fight or flight response to be activated. And as these muscles perform their function, they need nutrients to be brought to it. And guess what? The cardiovascular system is going to pump the blood to the muscles with the nutrients which were obtained from the digestive system. Now the blood is also going to contain gases like oxygen, which is brought in because of the respiratory system. The carbon dioxide which is generated in the muscles will be again removed by the respiratory system. Any of the waste products as a result of muscle contraction will be purified from the blood by the urinary system. And the blood will also contain some immune cells. You've heard of white blood cells, I'm sure. And along with the immune system, the integumentary system also plays an important role in protecting the body as well. So you can see how all of these various systems are working together. And on the last day of class, I will go through this slide again, and it should really make a lot of sense to all of you, even if it may have seemed overwhelming for the last 5 minutes. Okay, so do you have an understanding of how they all fit together and they work? Okay. So now let's talk about the various exams and how you can get an A in it. So in Unit 1, we'll be talking about the structure of water, proteins, energy metabolism, central dogma, organelles, and I think all of these topics, most of you may have had it in the prerequisite courses. And the new content may be the types of tissues, cell junctions. We'll talk about the integumentary and the skeletal system. So all of this will be in Exam 1. Exam 2 will be just the endocrine and the nervous system, and both of those can be pretty dense, so it makes sense that we only have these two systems in Exam 2. Exam 3 would be on the muscular system, energy, metabolism, cardiovascular system, blood, and the lymphatic and immune system. As far as Unit 4, you will have the respiratory system, digestive and urinary system, and the reproductive system, the last one, will actually not be in any of the first 4 exams. In fact, the final exam will be a cumulative final along with the reproductive system, and notice that all of these exams are worth 100 points, and if you add all of it, it would be 500 points. But guess what? We are going to drop the lowest unit exam score. So if something comes up and you can't take a single unit exam, you can drop it, but the final exam is mandatory. We can't drop that score. So in the unit exam, all of the unit exams will be in the classroom. The final will be in some other location. You will have 25 questions, each worth 4 points, and the final will also be worth 100 points. But notice that there are going to be 50 questions here, 30 from the first 4 units and 20 on the reproductive system, and each question is worth 2 points. So you may be wondering, if we have only 400 points, where do the remaining 100 points go to? And those are for the weekly lecture quizzes. So you see that the weekly lecture quizzes are due every Friday, and they all add up to 100 points. So we want you to be working regularly before the exams on these weekly quizzes. Please notice we don't round up the grade, and so if you get say 89.4 and you send us an email asking to round off your grade, we will send you this email back, right, saying that we are very sorry and we can't be able to do anything about it. And the reason I'm mentioning it now is now you can do something about it, and so I'm mentioning it now, right? So the weekly quizzes, you can access them through Canvas. So go click on the lecture quizzes on the home page. It will open up this page here on the right hand side, and notice that you will have 3 attempts for each of these quizzes, right? And the highest score on these attempts will be taken into consideration. But because we are pulling questions from an exam bank, you may not get the same questions on each of the attempts. So please review your notes before you take the lecture quizzes. And also there is an optional textbook. I know you already spent a lot of money to come to college, so we don't want you to buy another book. Everything you need for the exams will be presented in class, and so if you want, you can use this book. In terms of some general information, to access the PDF slides, PDF files of the slides, go click on lectures and you will find the respective PDF documents next to each lecture. And if you're not able to come to class, the lectures are live streamed on Echo 360, but if you want to see the updated subtitles, you'll have to access it within after 24 hours. And if you want to be in touch with us, please contact us through the course email address, because we will not be responding to messages on Canvas. All right, so that's all I want to mention now, and we should be able to respond in 24 hours to emails. Thank you all for your time, and I hope to see you tomorrow in class.